Welcome everybody today for our Prepare Institute seminar, and we have my friend Sumit Chanda here going to give our talk for today. Next slide. It's going to talk about building the plane while flying. Hopefully it's not a Boeing. Antiviral drug discovery in the time of COVID. He's a professor over at Scripps. Next slide. Principal investigator of his lab, which exploits systems-based approaches and cutting-edge technologies to study the virus-host interface and innate immunity. Has a focus on RNA viruses, those that are all out there to kill us with pandemic potential, and basic findings to drive drug discovery through Caliber and the translational research arm at Scripps, including the development of next-generation antivirals, adjuvants, vaccines, and immunotherapies, all those things that are going to save us all. Next slide. Oh yeah. Next seminar is in August. It's going to be pandemic prevention and human milk research, breast milk infectious disease tests. Here, the GFET aptamer biosensor, Sandra and Renish is going to be giving the talk. And Sandra just got a paper in PNAS, and it's great. So maybe we'll circulate that for the group. And then I'm going to let, I think you, Sumit, has the, you're going to have your slides that you can bring up.
Go first? How does that look?
Great. Just put it in there.
Yeah. So yeah, I think our pandemic response could have been designed by Boeing, kind of the way it rolled out. So what I'm here to do is hopefully convince you that we need to do a better job the next time around. But I'm going to start off by going back and telling you kind of our pandemic story and how our research unfolded back in what we like to call the before times. So can you guys see my pointer here?
Yep.
Yeah, okay, good. So I'm going to take you back to 2019. I had a postdoc in a lab who's now running his own lab over outside of Beijing. Xinyan went back to China on Christmas holidays, visiting family. He sent me a text message, said, hey, there's a pneumonia of unknown etiology outbreak in Hubei province in China. And I said, okay, keep me posted, see if they can figure out, you know, what's causing it. And he sent back basically a couple of days later, two-word text, just said novel coronavirus. And I think, you know, for most of us, when we hear, you know, novel influenza virus or novel coronavirus, and we're virologists, we start to really pay attention. So I asked him to kind of move back, get back to the US as quickly as possible, and we put together a team of virologists to see how we can start attacking this. And so at this time, things were only happening in China, and the virus was only accessible in China. So we reached out to two colleagues at Hong Kong University, Shofang Yuan and KY Yun. And then two other virologists, Ren Sun, who is at UCLA now at Westlake, and then Adolfo Garcia Sastra at Mount Sinai. And we started contemplating, okay, what can we do? And so we settled on reaching out to Arnab Chatterjee, who is at Caliber, which is a part of Scripps. And the idea was to screen this virus against their repurposing library. So there's a Reframe library. For those of you who don't know, one of the largest repurposing libraries available. They synthesized, they went through patent literature and synthesized about 6,000 compounds from scratch. The Gates Foundation gave them something like $20 to $30 million to put this together. It's accessible to anybody. So if anybody wants to screen it, I encourage you to reach out to Arnab and pitch that. So the idea was to send a postdoc in the lab, Laura Riva, to Hong Kong, because we knew how to screen. The folks in Hong Kong had the virus and knew how to grow the virus. Laura was going to go to Hong Kong with a suitcase full of this library and some automation. But in early February, Trump declared a travel ban, which meant that Laura could go to Hong Kong, but it was unclear if and when she would be able to come back. So that was definitely a no-go. So what we decided was to pivot and try to do drug discovery by iPhone. And so the first thing we did was we packed up everything that Laura was going to bring with her, sent it over to Hong Kong, which trying to ship in the middle of an epidemic. Of course, the package ended up in Guangzhou. Paul De Jesus is in the upper right here corner here, was on the phone with FedEx, I think, for 24 hours a day, making sure stuff was refilled with dry ice and so on. But things actually made it to Hong Kong. And so this is a picture of some of our automation here in their BSL-3 laboratory, got there all safe and sound. And so they use an app called WeChat, which is the Chinese equivalent of WhatsApp, to really kind of design and optimize and run this screen from halfway across the world. So Laura would be up in the middle of the night talking to Shofang and really kind of describing how to do what needs to be done as if she were there. So they had a burner phone in the BSL-3, and they just essentially FaceTime their way through this. So there's a nice article that Kim Tingley wrote about really what the heroics these guys went through here to do this. I encourage you to read it if you have some spare time. So the assay that we used, luckily, if you took SARS-CoV-2 and you put them on Vero cells, they induce the cells to die. So this is a common cytopathic effect of viruses. So we could easily measure viral replication by doing a live dead stain. Okay, probably not the most elegant readout, but at the time, it was the only thing we had, and so we went with it. So we took this reframed library of about 12 to 13,000 compounds. These are all known drugs with known safety and pharmacology. We spotted them, added Vero E6 cells, added the virus. This was a Hong Kong isolate at the time, waited for 72 hours and just asked, do any of these compounds protect from cell death or cytopathic effect? Okay. So we had several hundred hits come out of here. The nice thing about screening a known drug library is their MOAs are known, right? So we could take these drugs and figure out what these targets were and how they were modulating. So very few of these were direct-acting antivirals. Almost all of them were host-directed antivirals. We did pick up remdesivir on the screen, but that was people were already looking at that at the time. But things like endosomal proteases, signaling pathway members, nuclear transcription factors, mostly indirect effectors that were allowing the virus to replicate in these cells. And so by March of 2020, we were able to secure a shipment of the virus to be delivered to the lab. So this is Laura Martin getting the virus in what seems to be just like an Amazon shipment at the time. No masks, no gloves, nothing. Of course, in the BSL-3, she was completely suited up. And what she was able to do that was remarkable was start with 10 micromilliliters of virus and figure out a way to grow it up to the scale we needed, and also developed a secondary assay that we could directly look at virus replication. So we had an antibody from SARS-CoV-1 N protein, and she found that you could do a direct staining of infected cells to measure viral replication. And so instead of looking at this indirect effect of CPE, we could directly measure virally infected cells. So what we did was we developed a kind of a testing funnel here where we took the nearly 300 hits that came out of the screen, and then started adding more and more stringent criteria to this to try to get to things that could be then moved into humans for clinical testing, right? So we went from the CPE to the immunofluorescence, of roughly about a third confirmed in those. We went to then dose response. So we needed an EC90 and EC50 that we could hit because the pharmacology of these are known and the human plasma levels are known. We wanted to make sure that we could actually dose this in a human being at safe levels, at levels that would have antiviral efficacy. We looked at different cell lines to make sure that the cell lines weren't biasing things. We're looking at some mechanism of action, and I'll show you some of this data kind of going forward here. So this is the stage where we did the dose response and we got EC50s somewhere between kind of the double-digit nanomolar up to single-digit micromolar. On the left-hand side, you can see kind of the best hits that came out. So these are all things that we looked at, and they had EC50s that were and EC90s that were high enough that their Cmax and T half-life in humans that were previously reported could be achieved and still knock down the virus. So there are classes like cysteine proteases. I'll get into that in a little bit. Antivirals, I mentioned remdesivir. Chloroquines came out. Chloroquines, I think, famously or infamously, always do well in cell-based assays and never can be translated to in vivo or clinical. But nonetheless, this was a cell-based assay that they did read out. So some of our down selection criteria included moving into more relevant cell types. So remember, we started in vero cells. We went to iPS-derived pneumocytes. So here are 3 molecules. We started using remdesivir as a benchmark at that point. By that time, Gilead had been publishing extensively on remdesivir. Here's 3 other ones: Ono, MDL, and apilimod. All seem to work, and most of the drugs seem to work in these pneumocytes. These were particularly potent at reducing infection in the primary pneumocytes, which are the cell type that SARS-CoV-2 predominantly infects in the lungs. So Ono is something put out by Ono Pharmaceuticals, originally for osteoporosis. Here's the Cmax that's been previously reported, pretty good bioavailability. We're seeing an EC50 of about 410 nanomolar. It's a cathepsin inhibitor, right? So again, processing of the virus in the endosome through cathepsins, could have been surmised, but wasn't known at the time. It was nice to see that some of these cathepsin inhibitors, and here's another one. This is a cysteine protease inhibitor. It says it was targeted for a non-lysosomal, but we believe that in our case, that this is working through a lysosomal cysteine protease to cleave the spike protein and into its mature form, so the virus can escape the endosome and complete its replication cycle. The last kind of piece of confirmation we did was running these through in vivo lung tissue. So these are lung explants done in Hong Kong University, and we used one of our best molecules called apilimod, and you can see, depending on whether you look at intracellular RNA or plaque, it does as well as or better than remdesivir in this kind of more physiological setting. And what is apilimod? It is a PIK5 inhibitor, again, works on endocytosis. We had read reports that the company who owns apilimod took it into clinical trials. There were no further reports on clinical trials, so we assume that the clinical trials were not successful. Unclear as to why. But if they had worked, I'm sure we would have been able to, the company would have been promoting it fairly heavily. So we started to focus on another molecule called clofazimine. And so with remdesivir on the market, the reason we focus on clofazimine is that it's a known drug discovered in the 50s, FDA approved in 86 for the treatment of leprosy. It's cheap and it's orally bioavailable. And at the time where remdesivir was costing about $2,000 a treatment, and you had to go into the hospital or a medical facility to get it, getting a drug that could be expanded and cheap, and especially in the third world, places that could not get access to that, remdesivir would have been a pretty important thing. And so I'm showing you the dose response, and we went on to show that clofazimine works at two steps, both at entry and at RNA synthesis, and specifically, it was a hit inhibitor of the helicase function of SARS-CoV-2, and we could show this biochemically. And then working again with Shofang and KY in Hong Kong, we were able to show that both prophylactically and therapeutically, clofazimine reduced viral lung titers in mice. Not as good as remdesivir, but getting close. Also suppressed the inflammatory sequelae that really is an emblematic of severe disease. And if you look at this is the clofazimine treatment, this is the vehicle treatment, this is the mock infection, you can see that the alveolar sacs here are seem to be fairly well protected with clofazimine, and the vehicle, you see just the collapse and infiltration with immune cells here. So, but while we were doing this, a number of direct-acting antivirals started coming online. And so I mentioned remdesivir. It's an IV route, and one of the things with antiviral therapies is you need to onboard them fairly early in infection, or they lose their effectiveness. And so remdesivir wasn't doing great, but molnupiravir came online. The Pfizer drug Paxlovid. And then a series of antibodies that came online that seemed to work very well also. And unfortunately, many of these antibodies now, due to immune escape, no longer work. There's new ones coming online, but the appetite for these companies to develop new antibodies to kind of play whack-a-mole with the variants is becoming less and less. I think Lilly has now pulled out of this. And we'll see. AstraZeneca has a new one, but we'll see what the appetite is for pharma to develop these. Shionogi has a novel protease inhibitor also that looks pretty good, but they all have their liabilities, right? Whether it's genotox, Paxlovid, most of you know, has to be given with ritonavir, which is compounds drug-drug interaction problems, especially for people who are old age and take multiple drugs. It's a tough drug to kind of onboard concurrently. And so we continue to look for new SARS-CoV-2 antivirals, not only to just address the current variants of SARS-CoV-2 that are going around, but to make sure that we are prepared for a novel coronavirus-driven pandemic. And so I'm going to pitch the case to you why I think that these pandemics are no longer a 100-year event. I think many people were thinking, okay, 1918 Spanish flu, we had SARS-CoV-2, we're set for another 100 years. But I think this kind of timeline of pandemics makes it painfully obvious that these pandemics are coming, or viruses with pandemic potential are undergoing zoonotic transmission at a clip that has never really been seen before in human history, right? So what we used to consider a once in 100-year event, I think like hurricanes and forest fires, they're going to be coming once every 20, 25 years, right? And I think the slow-moving train wreck that we're watching right now is the bird flu, or H5 pandemic influenza. And so just the background and the story of a pandemic influenza. I mean, we've known about it, and it's been on our radar since '96, where there's an outbreak in China and Hong Kong, and this virus showed a 50% case fatality rate. Compare that to coronavirus, which had a less than a 1% case fatality rate. This is really something that I think is frightening, and more like the movie Contagion than the SARS-CoV-2 outbreak. So this has been really slowly drifting into kind of a different bird species. It's also genetically diversified. It's went from one continent to 3 to 4, and now it's in 6 continents. It's now been passed to mammalian cells, or to mammalian organisms. And as many of you know, in the US, it's been detected in goats and dairy cows. And there's been cow to human transmission. And there's been cases in the US, Europe, and Mexico now documenting animal to human transmission. We're one or a couple of mutations away from this virus, I believe, being able to be transmitted from human to human. And I think that's when the world again goes into that DEFCON mode, where we may have to lock everything down again. So why is this happening? So there are a number of factors: climate change, the habitat encroachment, and forest fragmentation. So both of these are changing the footprint where these reservoir species that harbor these viruses live. There's a lot more interconnectivity. People are flying everywhere, so it's much easier to transmit these viruses across continents. And then population densities, people are living closer and closer together. So that's the bad news. The good news is we have a pretty good idea of what the threat matrix is. I think besides for this disease X, the WHO coined term, we can see where the next threats could be coming from, right? The top 2 have always been novel influenza and novel corona, and then there's others, right? And so we can start to get prepared for these, right? We can start to develop vaccines, antivirals, and measures that protect us and buy us time until bespoke therapies and bespoke vaccines can be made to address directly the zoonotic strain that has emerged. And so one of the things that we're fortunate enough to participate in is a program from the NIH and NIAID called AVID. And it's a fairly large, about a $70 million grant to develop antivirals for coming pandemics. So Davey is on our SAB, and really have appreciated his input and service here. And I'm going to tell you a little bit about what this group has been able to accomplish. So this is kind of the all-star team for virology in my estimation. So this program is being led by me and Arnav Chatterjee at Scripps Research, and Adolfo Garcia Sastra at Mount Sinai. And again, just an incredible team of drug hunters, virologists, structural biologists, and other scientists that are really been able to push things forward at a clip that I think is truly amazing. So I won't go through all of it, but there's 4 projects focused on coronavirus, 1 project focused on Flavy, and the other focused on hemorrhagic fever virus. And I'm going to tell you 2 stories about what the advances they've made on targeting CL-Pro or M-Pro, which is a protease encoded in SARS-CoV-2, and then a polymerase inhibitor. And really not just to go back and treat SARS-CoV-2, but get broad spectrum activity. So if we had something else that popped out from a coronavirus, a bat coronavirus or an animated species, we would have something on hand that we could use to treat it. And so this MPRO inhibitor, the CL-PRO inhibitor, started from looking at hits that came out of the reframe screen. The folks at Caliber, the medicinal chemistry team, Elshan and Arnab Chatterjee here, made about 1,600 compounds and came up with a molecule called CMX 990. And this was more potent and better clearance than Paxlovid. The goal here is to get a once a day, maybe a twice a day, non-boosted, so no ritonavir and no ritonavir boost for SARS-CoV-2. And so far, this is looks like it has a pretty good chance of working. The PK and the tox looks really good. Right now, the PK has it at twice daily. We're trying to work on it to once daily. But if you look at in vivo, if we compare molnupiravir, vehicle control, the Pfizer drug, and our internal compound here, we're seeing attenuation of weight loss and suppression of viremia in the lungs. For a couple of outliers, we don't know, both for the Pfizer drug and our drug, we don't know where. We think these are experimental errors, but nonetheless, I think this is relatively convincing that it's working in vivo. And I think most compelling here is the spectrum of activity that we're seeing, right? So if you look at, this is the Paxlovid and this is the Shionogi, they're all pretty good at hitting SARS-CoV-2. This is a protease assay. But you start going into the other beta coronaviruses and other coronaviruses, you can start to see that the other protease inhibitors start to fall off. Whereas this C202 is just a derivative of that one compound. You can see that we're retaining activity all the way down to bird and whale gamma coronaviruses, right? And the deltas and the alphas all seem to be covered. So really, truly a pan-coronavirus protease inhibitor that can have us prepared for the next pandemic. A little shout out to Nick Heaton here, who did these protease assays. Next thing I want to share with you is a nucleoside analog that's been developed by Anil Gupta at Caliber as part of this program. So really, really impressive nuke, really can hit pretty much any respiratory virus that we've tried so far. So SARS-CoV-2, the seasonal coronaviruses, flu, rhinovirus, RSV, here are all the these are EC90s. So pretty potent against all of these. So imagine that you now have a drug that you can take anytime you get an upper respiratory tract infectant. If you ruled out bacterial, you can take this and not have to diagnose flu or RSV or HRV. So we're testing adeno, parainfluenza, and metapneumo. I'm not sure about adeno, but at least metapneumo and parainfluenza really have a broad spectrum respiratory virus therapy here in hand. It has even broader spectrum than that. Enteroviruses, flaviviruses, togaviruses, alpha viruses, really a remarkable novel nucleoside analog that has a much broader spectrum than you would see with remdesivir or other nuke analogs. So it does work in vivo. I've never seen a nucleoside analog work against flu, but you can see with this data here, MCOV 814 is a derivative, is a prodrug of 466. Flatlines titers of influenza. Similarly, for LCMV, below detection limit here, LCMV is our surrogate for LASA. And we're going to now use this data to test in a BSL-4 LASA model, and work by Mike Diamond shows that it can also suppress dengue virus replication. So really, I think we're on the heels of a broad spectrum that will help us prepare for nuke that can help us prepare for any almost any emerging viral infection going forward. And the last story I want to tell, this is my last slide, is work done in Juan Carlos de la Torre's lab by Rachel Sattler, who's a shared postdoc between me and Juan Carlos. And they're targeting the endonuclease domain of Lassa. So there's a drug called baloxavir, which targets the endonuclease domain of influenza. And the endonuclease is responsible for cap snatching for these viruses, which is a critical portion of the replication cycle. Baloxavir is a wonderful drug against flu. The Lassa endonuclease looks similar, but not the same. And so if you run, we use LCMV as a surrogate. If you run baloxavir in a replication assay, it doesn't do great. It does great in flu, single-digit nanomolar activity. Here we're pushed into single-digit micromolar EC50s. So we designed two analogs. So the Caliber MedChem team designed two analogs that drastically improves the EC50 of baloxavir against LCMV replication. There's a similar thing for Lassa when we tested against Lassa, and it also happens to have some activity against SFTSV and Hunin. It does have activity against flu, but it starts to really drop off from what we originally saw with the parental compound baloxavir. We're right now trying to finish off the PK studies, but we believe that we're in a dose range based on the preliminary PK data that we can have a once or twice daily treatment for Lassa fever and Hunin. And if the safety profile is anything like baloxavir, it should be an incredibly clean drug that really can offer the first treatment options for these hemorrhagic fever viruses. So that's the end of my talk. Just want to give a shout out to the folks that did all the work. Here's my lab, Laura Riva, Xin Yin, and my lab did the early coronavirus work. And here are the folks on the dream team there, Arnab, Shofang, KY, Adolfo, Andrew, and Ren. And then also want to acknowledge the folks at Caliber, Neil Gupta, with the and with the nuke work, Alshan on the Empro, Karen Case and Laura did a lot of the viral profiling. Arnab's kind of the head med chemist there and moves everything along. John Tajaro, Mike Diamond did a lot of the in vivo work, and the baloxavir work was done by Rachel Sattler with Juan Carlos de la Torre. So that's it for me. I'm not sure if we're doing questions right now, Davey, or wait till the next talk and then questions at the same time.
I think we can take some questions. Just an update on the Pilimod. They did look at it in hospitalized patients, which I think was, and I had said that at the beginning, that was a mistake. But
yeah, that's too late, right?
Yeah,
yeah,
exactly.
That's too late to do. Do you know if they did anything earlier for that?
No, they didn't do anything earlier.
Yeah.
It was
Yeah,
a
I think
mistake that
that's
was made often, honestly.
yeah.
People
They did that with remdesivir. They did that with a lot of these drugs, right? That
yeah.
When you have usually immune control of the virus by the time you end up in the hospital, right? So
Yep.
the antibodies they are of limited efficacy, right? And so I think turning towards the immunomodulators was a good idea, except I don't think we have good immunomodulators for this kind of viral immunopathology.
Yeah, I actually think that's going to be a really interesting question for what happens with flu when it hits, what immune modulators are going to be useful or not. People have always talked about steroids and nothing really panned out. And then when it happened with COVID, everybody, including myself, was anxious about using dexamethasone
Right.
with COVID because flu was always so tenuous. But yeah, that's what it worked. I wonder what's going to happen with bird flu. Anyway, I'll open it up. Does anybody have any questions? I don't think we have another talk today, so I think we got some
Oh, we
time.
don't? I could have gone longer. Sorry. I thought I only had 20
Yeah.
minutes.
I have a short attention span. It was great. Appreciate it. Anybody have any questions? Just feel free to chime in. I think that you might have covered everything. Um, this is great. We might have you back if you want to do something longer, or maybe you want to talk about bird flu. I am talking to the NIH now about what kind of clinical trial infrastructure we should be setting up to test things for bird flu. If you have, and we are looking for drugs, so if you wouldn't mind
Yeah.
helping
No.
me come
So,
up with a list, that
actually,
would be useful.
we
Okay.
should talk because, one of the programs we have going is trying to combine a baloxavir with a neuraminidase inhibitor, right?
Yeah.
Because what we saw with when we have resistance to both Tamiflu and baloxavir, it's not a huge deal because you're not seeing transmission, but once you kind of open up the playing field, right, like we saw with COVID, right, those resistant mutations start to become a problem. So one of the strategies we're taking is really to try to find a nice cocktail of drugs, right? With right now with baloxavir and Tamiflu, if we can add a third one to it, right, with a novel mechanism of action, maybe a nuke, I think that's really the way to go to corner the virus. I mean, we know this for Hep C and HIV, right? I think that we should start applying some of those lessons to the acute respiratory viruses.
Yeah, I agree. I think combo is the way to go. And that's how I'm designing it. So we'll see how um,
I have
it
a
all
really
falls
silly,
out.
can I ask a silly question?
Of course.
Go for it,
No,
Irene.
you got me all scared about the bird flu. So how will masks help with that until you guys figure out the drugs?
Well, the good thing is the drugs still all work on bird flu, right?
Okay.
And so I'm going to tell you a little secret. When I am in Mexico, I pop by and I pick up some oseltamivir, okay? And I have it, well, I'm not going to say where, so everybody doesn't show up. But both baloxavir and oseltamivir work. It's just we don't have enough, right? Oseltamivir is a pain to make. We don't have enough for the population. So if you're a first responder, you're probably pretty set. If I mean, I think an N95 mask will help, right? But it's until we know what that virus is, it's going to be hard to tell. But it wouldn't be a terrible idea to stock up on some Tamiflu. The stuff lasts forever, right? So you can take it whenever you as long as you want. But I mean, I think right now there's moderate concern. But what really worries me is that the surveillance apparatus isn't there, right? We don't have access to, they're not testing the farm workers and they're not, and so we don't know what's going on, and we're only going to know after the fact and when it's too late. And those first couple of weeks in a pandemic are the ones that matter the most, right? So if we had done something more in February, I think we could have saved tens of thousands, if not hundreds of thousands of lives. And so I think that's what's concerning right now to me, is that there's inadequate surveillance, and God forbid this thing ends up in pigs, right? If it ends up in pigs, I think that's when you go into your bunker and wave the white flag, right? Because I know the pigs are they're very much more like humans, right? And they're in the mixing vessels, right? So if you're going to see reassortments, right, that's probably where you're going to see the most reassortments from both swine influenza and other kind of avian, either high path or low path strains.
Okay. And we'll email you.
Sure.
Thank you.
Masks are more likely to work for flu than I think for at least Omicron of COVID. So Okay, this was great. Anybody else? All questions open. Margaret, do you want to, is limiting global mobility an effective response to pandemics? Well, it wasn't for COVID.
Yeah. Well, I
I don't
think,
think,
you can't shut down everything. And until you can't shut down everything, I think it's a little bit futile to do all of this. But, I think this is an important question, right? I mean, I get up and see the news clips of Fauci getting grilled in front of, Congress, right? Where their job should be trying to figure out what we did right and what we did wrong. We clearly did a lot of things wrong, and there's a lot of things we could have done better. But, instead of trying to figure out how you put Fauci in jail, I think a better use of taxpayer money is to try to, I mean, there should be like a 9/11 commission, right?
Exactly.
About what went wrong, what can we do better, right? Look at, the states that shut down harsh, look at the ones that didn't. You can look at Sweden, look, and come up with some, and I mean, you're comparing apples and oranges, but, I think there are ways to get lessons to what to do better the next time, instead of squabbling about, whether, Fauci funded some gain of function stuff in Wuhan Institute, right?
Crazy, crazy balls.
Yeah. I mean, just to follow up on that, I don't know if we've met in person, but I'm Margaret Hellworth. I oversee international services at Scripps. And, we became quite panicked, as other schools were, in trying to get the scientists here that we needed to get here. I remember Laura, actually.
Yeah.
And I just felt like a sort of blanket shutdown on global mobility, at least to me as a layperson, didn't make sense if we were still having people driving all between
Right.
states, especially people
Right.
flying from New York to other places where New York was really a hub
Yeah.
for the pandemic.
Yeah, and you had this thing where, okay, well, Americans can come back into the country, but not non-Americans,
Yeah.
right? I mean, somehow because Americans were not transmitters of the virus, right? I mean, none of the policies made sense, right? And I
No.
just don't think it's practical to do that again. And then, we're starting to see kind of the effects of the school shutdowns on the mental health of the kids. And I think you have to legitimately ask yourself, was it worth it, right? I mean, we know now that kids were not to say they weren't affected, but the case fatality rate in kids was significantly lower, right? And so, I think that there needs to be things put into place where once you measure kind of what the impact is on various populations, right? Let's have a plan. Let's have a playbook,
Mhm.
right? I mean, I'm sure there's a playbook for, if another plane flies into a building, God forbid, right? Where Homeland Security is ready, right? And look, it was terrible that 3,000 Americans died, but we had a Department of Homeland Security and we fought 2 wars because of it, right? 1 million Americans died here, and everyone wants to just forget about it, and they don't want to think about being prepared for the next pandemic, either because of pandemic fatigue, or they are laboring under the misconception that this isn't going to happen. This is a generational thing. I just think that at this point, and it's been so politicized, right, that I think it's difficult to get anybody to kind of pick up the mantle on this. But I think if we don't do it now, right, we're just doomed to repeat ourselves. And like I said, look, if bird flu 50%, that's probably an overestimation. I mean, I think that there were a lot of people who didn't report, but even if it's 10%, right, that's still 10 times worse than COVID, right? And you saw what COVID did to our kind of way of life, our healthcare system. I don't think that we will be able to sustain something that's 10 times worse than COVID.
Thank you.
yeah, the good news is that we do have some drugs that have some activity. I wouldn't say that Tamiflu or Baloxavir is all the best. So
No,
better
it's
drugs
something
needed.
though. Yeah, there's
it is.
something, right? There's something that will buy us some time, right? And we know how to make good vaccines against flu, or decent vaccines against flu. You know, whereas we had to kind of start from scratch for coronaviruses. But you know, it's the scaling up that takes months and months, right? We don't have, you know, I think most people imagine we have these huge stockpiles where we can give away these things that we don't, right? We have stockpiles for first responders and military. I think the rest of us are kind of on their own. So, you know, there just needs to be, I think, a concerted, comprehensive strategy to look at kind of what the threat horizon is and what are we going to do to better prepare ourselves so we don't have to go through the same thing we went through the last time.
Okay, this has been great. Any other
Great.
questions? I think we're good.
Okay,
Thanks,
great.
thanks
Thanks,
to
Davey.
everybody.